Hello, good morning friends. Today I talk about you Bioinformatics Blast. Blast is a basic local alignment search tool has become the de facto standard in search and alignment tools. The Blast algorithm is still actively being developed and is one of the most cited papers ever written in this field of biology. Many researchers use BLAST as an initial screening of their sequence data from the laboratory and to get an idea of what they are working on. BLAST is far from being basic as the name indicates. It is a highly advanced algorithm which has become very popular due to availability, speed, and accuracy. In short, a blast search identifies homologous sequence by searching one or more databases usually hosted by Ncbi on the query sequence of interest. BLAST is an open source program and anyone can download and charge the program code. This has also given rise to a number of BLAST derivatives. WU BLAST is probably the most commonly used. BLAST is highly scalable and comes in a number of different computer platform configurations which makes uses on both small desktop computers and large computer clusters possible. Example of BLAST uses. BLAST can be used for a lot of different purposes. A few of them are mentioned below. Looking for species. If you are sequencing DNA from unknown species, BLAST may help identify the correct species or homologous species. Looking for domains. If you blast a protein sequence or a translated nucleotide sequence, BLAST will look for known domain in the query sequence. Looking at phylogeneity, you can use the BLAST web page to generate a phylogenetic tree of the BLAST result. Mapping DNA to a known chromosome. If you are sequencing a gene from a known species but have no idea of its chromosome location, BLAST can help you. BLAST will show you the position of the query sequence in relation to the hit sequence. Annotation. BLAST can also be used to map annotation from one organism to another or look for common gene in two related species. Searching for homology. Most research projects involving sequencing of either DNA or protein have a recruitment for obtaining biological information of the newly sequenced and maybe unknown sequence. If you if the researchers if the researchers have no prior information of the sequence and biological contents, valuable information can obtain be often be obtained using BLAST. The BLAST algorithm will search for homologous sequence in predefined and annotated database of user's choice. In an easy and fast away the researchers can gain knowledge of gene of protein function and find evolutionary relations between the newly sequenced DNA and well-established data. After the blast search, the user will receive a report specifying found homologous sequences and their local alignment to the query sequence. How does BLAST work? BLAST identifies homologous sequence using a heuristic method which initially finds short matches between two sequences. Thus, the method does not take their entire sequence space into account. After initial match, BLAST attempt to start local alignment from these initial matches. This also means that BLAST does not guarantee the optimal alignment. Thus, some sequence hits may be missed. In order to find optimal alignment, the Smith-Waterman algorithm should be used. In the following, the BLAST algorithm is described in more detail. Seeding. When finding a match between a query sequence and a hit sequence, the starting point is a word that the two sequences have in common. A word is simply defined as a number of letters. For BLAST P, this default word size is 3 W equal 3. If a query sequence has a QWRTG, the search words are QWR, WRT, and RTG. See Figure 1. During the initial blast seeding, the algorithm finds all common words between the query sequence and the hit sequence. Only region with a word hit will be used to build an alignment. Which BLAST program should I use? Depending on the nature of sequence, it is possible to use different BLAST programs for the database search. There are five versions of BLAST program: BLAST P, BLAST N, BLAST XT, BLAST N, and T-BLAST X. BLAST N, query type nucleotide or database type nucleotide, comparison nucleotide to nucleotide. BLAST P, query type protein, database protein, comparison protein to protein. T-BLAST N, query protein, database type nucleotide, comparison protein to protein. BLAST X, query nucleotide but database type protein. Comparison protein-protein. The query are translated into protein. T-BLAST-X, nucleotide query and database type nucleotide. Comparison protein-protein. The query and database are translated into protein. The most commonly used method is BLAST, a nucleotide sequence against a nucleotide database, or a protein sequence against a protein database, but often another BLAST program will produce more interesting hits. Example, if a nucleotide sequence is translated before the search, it is, it is more likely to find better and more accurate hits than just a BLAST and search. One of the reason for this is the protein sequence are evolutionary more conserved than nucleotide sequence. Another good reason for translating the query sequence before the search is that you get protein hits which are likely to be annotated. Thus you can directly see the protein function of the sequence gene. Which BLAST option should I change? The Ncbi BLAST web page can the BLAST command line tool offer a number of different options which can be changed in order to obtain the best possible result. Changing these parameters can have a great impact on the search result. It is not the scope of document to comment on all of the options available, but merely the options which can a change with a direct impact on the search result. What is e-value? The impact value. E-value is the impact value. Can be changed in order to limit the number of hits to the most significant ones. The lower of e-value, the better than hit. The e-values depend on the length of the query sequence and the size of database. For example, an alignment obtaining an e-value of 0.05 means that there is a 5 in 100 chance of accuracy by chance alone. E-value are very depending on the query sequence length and the database size. Short identical sequence may have a high e-value and may be regarded as a false positive hits. This is often seen